The sensory system. An introduction to the sensory systems. Ask anyone what the senses are, and they are likely to visit the 5 major senses as taste, smell, touch, hearing, and sight. However, these are not all of the senses. The most obvious omission from this list is balance. Touch can be further subdivided into pressure, vibration, stretch, and hair follicle position on the basis of the type of mechanoreceptors that perceive these touch sensations. Other overlooked senses include temperature perception by thermoreceptors and pain perception by nociceptors. Within the realm of physiology, senses can be classified as either general or special. A general sense is one that is distributed throughout the body and has receptor cells within the structure of other organs. Mechanoreceptors in the skin, muscles, or the walls of blood vessels are examples of this type. General senses often contribute to the sense of touch, as described above, or to proprioception and kinesthesia, or to a visceral sense, which is most important to autonomic functions. A special sense is one that has a specific organ devoted to it, namely the eye, inner ear, tongue, or nose. Gustation is taste and olfaction smell. Gustation, taste. Gustation is the special sense associated with the tongue. The surface of the tongue, along with the rest of the oral cavity, is lined by a stratified squamous epithelium. Raised bumps called papillae contain the structures for gustatory transduction. There are 4 types of papillae based on their appearance: circumvallate, foliate, filiform, fungiform. Within the structure of the papillae are taste buds that contain specialized gustatory receptor cells for the transduction of taste stimuli. These receptor cells are sensitive to the chemicals contained within foods that are ingested, and they release neurotransmitters based on the amount of the chemical in the food. Neurotransmitters from the gustatory cells can activate sensory neurons in the facial, glossopharyngeal, and vagus cranial nerves. Only a few recognized submodalities exist within the sense of taste or gustation. Until recently, only 4 tastes were recognized: sweet, salty, sour, and bitter. Research at the turn of the 20th century led to recognition of the 5th taste, umami, during the mid-1980s. Very recent research has suggested that there may also be a 6th taste for fats or lipids. Olfaction, smell. The human body can detect over 10,000 odors. Like taste, olfaction is also responsive to chemical stimuli. The olfactor receptor neurons are located in a small region within the superior nasal cavity. The nasal epithelium, including the olfactory cells, can be harmed by airborne toxic chemicals. Scent receptor messages travel to the cerebrum, specifically to the primary olfactory cortex that is located in the inferior and medial areas of the temporal lobe, and additionally to the hypothalamus, where smells become associated with long-term memory and emotional response. Audition or hearing, equilibrium or balance, and somatosensation or touch. Hearing or audition is the transduction of sound waves into a neural signal that is made possible by the structures of the ear. The external ear consists of the auricle, sometimes referred to as the pinna, ear canal, and tympanic membrane. The C-shaped curves of the auricle direct sound waves toward the auditory canal. The canal enters the skull through the external auditory meatus of the temporal bone. At the end of the auditory canal is the tympanic membrane, which vibrates after it is struck by sound waves. The middle ear consists of the ossicles, oval window, and tympanic membrane. 3 ossicles are the malleus, incus, and stapes, which are Latin names that roughly translate to hammer, anvil, and stirrup. The malleus is attached to the tympanic membrane and articulates with the incus. The incus, in turn, articulates with the stapes. The stapes is then attached to the inner ear, where the sound waves will be transduced into a neural signal. Vibrations of the ossicles travel through the oval window, moving fluid in a wave-like motion. The frequency of the fluid waves match the frequencies of the sound waves. The middle ear is connected to the pharynx through the eustachian tube, which helps equilibrate air pressure across the tympanic membrane. The tube is normally closed, but will pop open when the muscles of the pharynx contract during swallowing or yawning. The inner ear is often described as a bony labyrinth, as it is composed of a series of canals embedded within the temporal bone. It consists of the cochlea, that is responsible for hearing, and the vestibule, that is responsible for balance. The neural signals from these 2 regions are relayed to the brainstem through separate fiber bundles. However, these 2 distinct bundles travel together from the inner ear to the brainstem as the vestibulocochlear nerve. Sound is transduced into neural signals within the cochlear region of the inner ear, which contains the sensory neurons of the spiral ganglia. These ganglia are located within the spiral-shaped cochlea of the inner ear. The cochlea is attached to the stapes through the oval window. The image below is a cross-sectional view of the cochlea that shows the scala vestibuli and scala tympani, which run along both sides of the cochlear duct. The cochlear duct contains several organs. The organs of corti, which transduce the wave motion of the 2 scala into neural signals. The organs of corti lie on top of the basilar membrane, which is the side of the cochlear duct located between the organs of corti and the scala tympani. As the fluid waves move through the scala vestibuli and scala tympani, the basilar membrane moves at a specific spot depending on the frequency of the waves. Higher frequency waves move the region of the basilar membrane that is close to the base of the cochlea. Lower frequency waves move the region of the basilar membrane that is near the tip of the cochlea. The cochlea encodes auditory stimuli for frequency between 20 and 20,000 hertz, which is the range of sound that human ears can detect. The unit of hertz measures the frequency of sound waves in terms of cycles produced per second. Frequencies as low as 20 hertz are detected by hair cells in the apex or tip of the cochlea. Frequencies in the higher ranges of 20 kilohertz are encoded by hair cells at the base of the cochlea, close to the round and oval windows. Most auditory stimuli contain a mixture of sounds at a variety of frequencies and intensities, represented by the amplitude of the sound wave. The hair cells along the length of the cochlear duct, which are each sensitive to a particular frequency, allow the cochlea to separate auditory stimuli by frequency just as a prism separates visible light into its component colors. Sound travels at a speed of 1,130 feet per second.